let's talk about what goes on at the single cell level. How does a gene for influenza hemagglutinin get into a human virus? And I've prepared a small video to show you how this goes. And before we roll the tape, let me just introduce you to the fact that I'm going to show two influenza strains here, one of which has red genomic segments and one blue. Okay? And what you're going to see in the video is that new human hemagglutinin types come about by recombination between two strains. And that recombination requires that the two different strains infect the same cell. And when they do that, what you're going to see are the two viruses binding to the cell surface receptors as before, enter the cytoplasm, disassemble, and start replicating their genomes. And then you're going to see an important new event happen. All right, so let's roll the tape. And here you can see particles are going to bind to the cell surface and fuse with the plasma membrane, release their contents, disassemble, and liberate their genomes. Now, let's stop the tape for a second. You know from last time that the next event that's going to happen in viral replication is that these RNA segments are going to be, first of all, they're going to be transcribed and translated to generate more viral proteins. The RNA is going to undergo RNA-dependent RNA replication, so you're going to get lots of new copies of the subunit. But now, because two different strains have infected the same cell, their RNA subunits are going to freely admix with one another, so that when the time comes late in infection to reassemble into daughter virus particles, subunits from one strain can occasionally be picked up along with subunits of the other strain and packaged into a single virus particle. So let's roll the tape and watch that process happen. That is a form of recombination that results from the mixing of subunits, and you'll see it shortly. So here are the newly replicated genomes, and they're going to go out now and assemble with newly made proteins into progeny particles. And here you can see the process happening. And if you just concentrate on this particle, you can see, let's stop the tape, that two blue subunits have been incorporated along with 6 red subunits, and vice versa over here. And you can see that from such a dually infected cell, a whole host of new recombinant viruses bearing different subunits have occurred. So this is a form of genetic exchange that occurs not by crossing over, but by swapping whole viral mini chromosomes, if you will. Okay? And so we can complete the tape now, and you'll see these particles will go out, pick up their envelopes, and go off into the outside world. This will be a mixed stock with all different varieties of recombinants, and those recombinants that do best in nature will survive. Okay, so that at the molecular level, and we can terminate the tape now, is where new recombinant strains come from. That is, at the cellular and molecular level, the ultimate source of all these new shifted variants.